Welcome to an exploration of one of cooking's most fascinating mysteries, the vinegar paradox. Why does a tiny splash of vinegar make fruit taste sweeter while too much completely destroys the flavor? Let's uncover the science behind this culinary phenomenon. First, we need to understand the fundamental chemistry of how our taste receptors work. The human tongue contains 5 primary taste receptor types: sweet, sour, salty, bitter, and umami. These 5 taste receptors work together to create the complex flavor profiles we experience in food. Vinegar contains acetic acid, which dissociates into acetate ions and hydrogen ions in solution. These hydrogen ions are the key players that activate our sour taste receptors, creating that characteristic tangy sensation. Understanding pH is crucial to explaining the vinegar paradox. The pH scale ranges from 0 to 14, with acidic substances below 7, neutral at 7, and basic above 7. pH is defined as the negative logarithm of hydrogen ion concentration. This logarithmic scale means each pH unit represents a 10-fold change in acidity. To put this in perspective, lemon juice has a pH around 2, tomatoes around 4.5, and milk around 6.5. Vinegar typically has a pH between 2.5 and 3.5, making it quite acidic. Now let's explore why small amounts of acid enhance sweetness and fruitiness. Here's an interesting question. Why would adding something sour make sweet things taste even sweeter? It seems counterintuitive, doesn't it? This graph shows perceived sweetness versus acid amount. Notice the enhancement zone where a small amount of acid actually increases the perception of sweetness. The answer lies in sensory contrast and receptor interaction. Our taste system uses comparison to judge intensities. We can model this as perceived sweetness equals baseline sweetness times 1 plus alpha times acid concentration, where alpha is a small enhancement factor. For small acid amounts, this enhancement factor alpha is approximately 0.3, meaning a 30% boost in perceived sweetness. Let's look at a concrete example. Imagine we have 10 grams of sugar dissolved in a dessert. Adding just half a gram of vinegar to this creates a 30% boost in sweetness perception. The dessert tastes like it contains 13 grams of sugar instead of 10. Let's dive deeper into the molecular mechanism behind this enhancement effect. 3 key mechanisms work together to create the enhancement effect. First, sensory contrast. Second, saliva production changes. Third, volatile aroma compound release increases. Saliva flow rate increases proportionally to hydrogen ion concentration raised to the 0.4 power. More saliva means better distribution of taste molecules. When acid drops the pH in your mouth, it increases the release of volatile aromatic compounds from the food. The volatility factor can be expressed as 10 raised to the power of 7 minus pH divided by 3. Lower pH means higher volatility. At pH 6, the volatility factor is about 3.2. At pH 4, it jumps to 10.0, meaning 3 times more aromatic compounds reach a nose. Now we reach the critical part. What happens when we add too much acid? At what point does the beneficial effect reverse, and acid starts destroying flavor instead of enhancing it? This extended graph shows the complete picture. After the enhancement zone, there's a sharp decline into the destruction zone, where flavor perception collapses. The answer is around 2 to 3% acetic acid concentration by weight in the final dish. Beyond this, taste receptors become overwhelmed. In molarity terms, the critical point occurs around 0.35 molar acetic acid concentration. 3 destruction mechanisms activate when acid exceeds this threshold. First, receptor saturation occurs. Second, pain receptors activate. Third, sensory fatigue sets in. Let's examine receptor saturation in detail, as it's the primary destruction mechanism. Taste receptors bind hydrogen ions in a reversible reaction. Receptor plus hydrogen ion forms the activated receptor complex. The signal follows Michaelis Menten kinetics. Signal equals hydrogen ion concentration divided by the dissociation constant plus hydrogen ion concentration. At low acid concentrations, the signal increases linearly with concentration. Each additional hydrogen ion produces more signal. At high acid concentrations, receptors become saturated. The signal plateaus because all receptors are occupied, and adding more acid produces no additional signal. This saturation curve shows how receptor response flattens at high concentrations. The red line indicates maximum signal capacity. The dissociation constant for sour receptors is approximately 0.01 molar. When hydrogen ion concentration exceeds this by tenfold, saturation occurs. When sour receptors are saturated, they dominate the signal sent to your brain, and you cannot effectively detect sweet or fruity notes anymore. The second destruction mechanism involves pain receptors, not taste receptors. High acidity activates specialized pain receptors called TRPV1 channels and acid-sensing ion channels, or ASICs. The pain threshold occurs when pH drops below approximately 3.5 in most individuals. Going from pH 4 to pH 2.5 crosses the pain threshold. Pain signals override and mask delicate flavor signals. Pain signals are prioritized by your nervous system. When pain receptors fire, they mask all other taste perceptions as a protective mechanism. We can model the masking factor as 1 minus E to the power of negative pH threshold minus current pH divided by 0.5. At pH 3, the masking factor is 0.4, meaning you lose 40% of your ability to perceive other flavors. At pH 2, the masking factor jumps to 0.9, resulting in nearly complete flavor loss. All you taste is burning sourness. Let's apply this knowledge to real cooking scenarios with specific calculations. Example 1. A fruit salad dressed with balsamic vinegar. This is a perfect enhancement scenario. We have 500 grams of strawberries and 10 grams of balsamic vinegar, which contains about 6% acetic acid. The acid mass is 10 times 0.06, which equals 0.6 grams of acetic acid. The concentration in the final dish is 0.6 divided by 510 total grams, which is approximately 0.12%. This concentration is well within the enhancement zone. The strawberries will taste sweeter, and their fruity notes will be amplified. Before the vinegar, you have normal sweetness. After, you get a 30% enhancement in perceived sweetness plus amplified berry aromas. Example 2. An overly vinegary salad dressing where someone added too much vinegar. We have 100 grams of salad with 30 grams of regular vinegar, which is 5% acetic acid, in the dressing. The acid mass is 30 times 0.05, which equals 1.5 grams. The concentration is 1.5 divided by 130, approximately 1.15%. At 1.15%, we're approaching the destruction zone. The salad will taste unpleasantly sour, masking the freshness of the vegetables. The fresh vegetable flavors get overwhelmed by harsh, overpowering acidity. Based on all this science, what are the practical guidelines for using acid in cooking? The enhancement zone is 0.05 to 0.5% acetic acid. The transition zone is 0.5 to 1.5%. Above 1.5%, you enter the destruction zone. For a typical 500-gram dish, enhancement requires just 0.25 to 2.5 grams of acetic acid. To calculate vinegar amount, divide the acid needed by 0.05, which is the typical acetic acid concentration in vinegar. For example, to add 1 gram of acid, you need 20 grams, or about 4 teaspoons of regular vinegar. Temperature significantly affects the vinegar paradox in ways you might not expect. Hot dishes amplify sour perception because temperature increases the activity of taste receptors and volatile release. Sour intensity is proportional to temperature raised to the power of 1.3. A 20-degree increase can boost sour perception by 40%. This graph shows sour perception intensity versus temperature. The curve rises sharply as temperature increases. Conversely, cold dishes need more acid to achieve the same enhancement effect because receptors are less sensitive when cold. Ice cream can handle 0.8% acid concentration and still taste balanced, while hot soup can only handle 0.3% before becoming too sour. The presence of other ingredients affects how acid behaves through chemical buffering. Proteins and minerals in food act as pH buffers, absorbing some of the added acid and moderating pH changes. Buffer capacity is defined as the change in acid needed to produce a unit change in pH. Dairy products have high buffer capacity due to proteins and calcium, while fruits have low buffer capacity. Dairy on the left shows high buffer capacity with a large pH-absorbing volume. Fruit on the right shows low buffer capacity with minimal absorption. This buffering explains why you can add much more acid to yogurt than to fruit juice before the sourness becomes unpleasant. The effective hydrogen ion concentration equals added hydrogen ions divided by 1 plus the buffer capacity beta. Let's synthesize everything into practical cooking wisdom you can use immediately. Rule 1. Start with 0.2% acid concentration and adjust upward by taste. For a 1 kilogram dish, that's just 2 grams of acid or 40 grams of vinegar. Rule 2. Add acid at the end of cooking. Heat degrades some acids and changes perception, so final adjustment is crucial. Rule 3. Account for existing acidity in your ingredients. Tomatoes already contain citric acid, so they need less added vinegar. This bar chart shows how different ingredients require decreasing amounts of added acid based on their natural acidity levels. Rule 4. Use the right type and amount of acid for the job considering the specific ingredients, desired flavor profile, and cooking method. For example, citric acid is great for fruits and desserts, while acetic acid is better suited for savory dishes.